drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a Ph.D. in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. If you're getting value from this content, please consider becoming a paid subscriber to the Mind and Matter Substack. For just $5 a month, you'll get early access to episodes and other content, full episode transcripts, and I'll prioritize answering your questions in emails and the comments sections. You can also support me by checking out the links in the episode description to my affiliate partners who make various health and wellness products that I use and enjoy myself. You'll receive a discount and help support the podcast that way. And if you want the benefits of a paid subscription, but it's not in your budget, simply sign up for my free weekly newsletter, send me an email, and I'll give you a complimentary paid subscription. Word of mouth is the best way to help the podcast grow. So if you like what I'm doing, please share your favorite episodes with family and friends. The purpose of the podcast is to provide you with information obtained largely from primary sources, the people doing the research and producing new knowledge. This content is never meant to serve as medical advice. And with that, enjoy the episode. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.
I come from Italy, so this is where I did my bachelor's and my master's. I am a biologist, and during my master's, I switched to neurobiology because I was very intrigued by how the brain works, and really the question that drove my studies and my formation was this very existential question of like what makes us who we are. And so I thought that by understanding our body and our mind, I could get a little bit closer to that question. And so after the end of my master in neurobiology, where I still had like a very strong background in basic molecular biology and neuroscience, then I decided to join the lab of Johannes Graf for my Ph.D. because I had these two main curiosity, one being epigenetics and the other one being memory. And so in his lab, I was like, okay, can I do like a project where I can combine both fields? And he was super cool because he said, yeah, of course, I'm also very interested in these two aspects of science. And so we had a lot of fun over the last years, like trying to figure out how to study epigenetics and memory at once.
So on the question of what makes us who we are, one thing we can talk about is what makes a cell, what a cell is. So obviously we start from a single cell, and then the embryo develops and we go from one cell to many, many, many cells, and there's many different cell types. Right? There's neurons, there's skin cells, there's different types of neurons, there's different types of skin cells, and so on and so forth. At a very basic level, what actually differentiates one cell from another? They all have the same genome. So what determines if something's, a neuron type A, neuron type B, and so forth?
This when I first understood this, it really blew my mind, because essentially almost every cell in our body has this 2 meters long stretch of DNA inside. And so, as you well said, the DNA is equal in all cells, and yet we have such a massive diversity. And this is because this 2 meters long DNA has to be wrapped and condensed in an environment that is microscopic. And this implies that during the wrapping, the conformation will make it so there will be parts of the genome that are totally inaccessible, so where the chromatin will be very condensed, and whereas other parts, other regions will be more accessible. The DNA over these parts is more relaxed, and therefore the genes that are left in these more open, accessible regions will be the genes that characterize the identity and the functions of that cell. And this is what epigenetics stands for in a very macroscopical sense, because it's really like allowing for this three-dimensional conformation of the DNA into such a small space, like the nucleus.
I see. So there's enough DNA in our cells that if it was just stretched out linearly in a straight line, you'd be talking about meters of DNA. And yet somehow all of that has to fit inside of every single one of our cells. And so that DNA is bound up or wound up very tightly. And I guess a picture that could offer an analogy here would be like a fishing pole. You've got a lot of line, but it has to like wrap very tightly in one little space. And so based on which length of the DNA segments are available or unwrapped, that determines which genes could be used by the cell to make proteins.
Exactly. Yes. And of course what the analogy you used is very, very good. There is also this analogy sometimes of the necklace of some ladies with pearls. So essentially these proteins you so well introduced are called histones, and they really serve to wrap DNA around it. And what is interesting is that these histones are not fixed. I mean, they can be displaced, they can be changed into different variants depending on many different factors. And so there are some regions that we know are called constitutively open regions. So those usually bear genes that are really making up the most of a cell, the most of the identity of a cell. Whereas there are other regions that are more variable. They can be open, they can be closed. And this really is more what determines the function of that cell. And the epigenetic factors that are like essentially epigenetic factors like chemical groups. So we have so many different chemical groups that can be harbored on the tails of these histones, and depending on the combination, and some people refer to a epigenetic code because they can be so many different and also organized in so many different loci of the histone tails of different amino acids. So depending on the code that they will compose, then some codes will promote more openness and therefore more accessibility for the genes that are found around those histones to be read and then expressed, whereas some codes instead promote more closure. And so even though these regions can be accessible, it doesn't mean that they will be accessible for that specific cell at that specific moment.
Mm-hmm. So it sounds like the openness of the DNA, the interaction between the DNA and the histone proteins around which the DNA is wrapped, there's got to be a balance here, because on the one hand, you don't want all of the DNA available and turned on, because then you wouldn't be able to maintain the identity of a cell. So a neuron wouldn't be able to stay a neuron. if everything just turned on. But on the other hand, you can't have everything completely static. You have to have some room to turn some genes on and off, because that's what's ultimately going to enable that cell to change somewhat. So in the context of the nervous system, it could build stronger synapses, it could get rid of synapses. Neuroplasticity would require that some of these genes can turn on and off to some extent, while at the same time maintaining the overall identity of the cell.
Yeah, exactly. And this is the beauty of epigenetics as well, because there are like these chemical groups that are added on these histones, or for example, methylation can also be added on the cytosine groups of the DNA. So this chemistry can be modified by several classes of enzymes. We have erasers and we have writers that are really found at these dichotomous, they have this very opposite role. So while the writers, they really add different chemical groups on this amino acid found on the histones, the erasers, on the other hand, they will remove these chemical groups. And so this really talks in favor of this balance in a way, because the system somehow, by balancing the activity and the content in the cells of these classes of enzymes, it will therefore balance the presence of the chemical groups on the histones, and therefore the accessibility or not of the chromatin to transcriptional machineries.
Mm-hmm. And so you've got, so what are some of these reading and writing capabilities? So you mentioned DNA methylation, so you can add a little chemical group to certain letters in the alphabet of the DNA. What are some of the other mechanisms there that do this reading and writing?
So in terms of methylation, is this one special mark, because as you said, can be present in the DNA and as well on the histone tail. So in terms of histone post-translational modification, so those are called like these chemical groups added on the histone tails, we have methylations. In my personal research, I focus particularly on histone acetylation. Because it's really like a splendid tool to promote openness of the DNA, of the chromatin structure, because histone acetyl groups, they manage to, by repulsion, really stretch and open and relax the chromatin and make it accessible. But there is also phosphorylation right now. I mean, we have, this is really a wonderful time to be studying epigenetics, because there are so many new PTMs, so post-translational modifications being discovered every day. There is serotonylation, lactosylation, and all these new chemical groups seems to also contribute to the overall three-dimensional organization of the DNA in the nucleus.
I see. So you said post-translational modification. So this would mean that proteins get made within the cell. So histones would be one example of a protein, where it's a sequence of amino acids that's got a shape to it. And after the protein is actually made, after it's manufactured, translated, you can modify it with various chemical residues. So the one that you mentioned was acetylation. So the idea is just there's proteins in the cell, they have an amino acid sequence, they have a three-dimensional structure, but then you can modify them after that, after they're already made and functioning, and that can determine things like the DNA winding.
Yeah, perfect. Yeah, it's like a Christmas tree, you have your histone, it's like the bare Christmas tree, and then you can add all sorts of stuff on top.
And so what's the relationship between acetylation and transcription, or turning genes on? Which one?
The acetylation is one of these modifications that we decided to study in the context of memory, because it has been associated in the nucleus to neuronal activity, for example. So this histone modification is very interesting because it is linked to the energy levels of a cell. These acetyl groups, they derive from, for example, acetyl-CoA, which is one of these byproduct of the glycolysis or of many other metabolic pathways, actually. And so whenever you have this pool of acetyl-CoA, they kind of somehow correlate with the amount of acetylation you would have in the nucleus. And this is interesting because we see, a neuron has to use a lot of energy to fire and to function. And therefore, we see that, like, for example, after let's call it synthetic neuronal activation or chemically induced neuronal activation, for example, sometimes we do experiments in petri dishes where we cultivate and grow neurons. And there, if you use specific chemical compounds, we can induce neuronal activity. And this has been shown both in vitro system or also in vivo using mice and stimulating them both either with chemical compounds or with like behavior, like for example, letting the mice exploring a new arena or for example, learn something new. So both the chemical induced neuronal activation as much as the natural induced neuronal activation is correlating with higher histone acetylation levels. And these high histone acetylation levels have been associated to higher level of gene transcriptions. And there have been plenty of studies really showing how by promoting histone acetylation, you really relax the portions of the genes called promoters, as well as enhancers that are more like distal regulatory regions of the genome that really manage to go to the promoter and carry and complete these transcriptional machineries that will be essential for allowing robust gene expression. And yeah.
I see. So cells have actually they've coupled their bioenergetic supply of fuel in the form of its ability to make ATP to its potential for doing gene transcription through acetylation. Because so you mentioned that these histones become acetylated, so you added an acetyl group to them. This enables more genes to be turned on. That's a very energetic process inherently, right? You have to use proteins to transcribe the genes. You've got to fold the proteins, then you've got to shuttle them to maybe the far end of a neuron, for example. That requires a lot of fuel. So it sounds like there's sort of something elegant going on here where the actual signal that opens up or that modifies the histones to open up the potential for transcription is actually coming from acetyl-CoA itself, which is basically the precursor to make ATP in the cell.
Yeah, as I said, I think right now we are still at the correlative level. I think in other studies, more like fundamental biology studies on yeast, for example, they really investigated deeper the role and contributions of metabolism to gene expression and epigenetics profiles. In neuroscience, we are still not at this like causality level, but I think that there are plenty of studies showing how like metabolism really supports neuronal activity and really like defines the type of epigenetic landscapes that you have. So for sure, like knowing that the building blocks, so these bricks of acetylation derive from metabolism, even though we are still lacking the precise experiments to test how much and from which pathway then would change which regions of the genome, we have a pretty solid background to really rely on and say, yeah, for sure they are linked.
And I want to give people a sense for the basic steps involved in, say, doing something like building a synapse or the connection between the nucleus of a neuron and the synapse of a neuron. So neurons, they're sort of a weird cells in many ways. They've got a weird shape. A synapse or a connection between a given neuron and one of its partners might be very, very far away from the nucleus of that neuron's where the DNA is. And so, when we're talking about transcription and turning genes on and off, the genes are in the nucleus. They have to be turned on. They have to be made into proteins. But then, if you want to build a synapse, for example, you've got to shuttle that protein all the way, quite a distance to the synapse. And that probably takes a lot of machinery and a lot of energy. So can you give us a sense for how expensive and how costly it is to do stuff in the nucleus and then ship it all the way to the synapse?
So I just want to make a distinction because we know that not all the synapses need to. So for sure, let's say if we would start to build a neuron from scratch, then the nucleus would be fundamental. But let's say once the neuron is already set up, there will be, it is already somehow connected, and there are already some points, like with some junctions between neurons. So there are synapses that are constantly like maintained at the synaptic level, because in the synapse there are still like ribosomes, sometimes even mRNA, so which is usually this like stretch of messenger RNA that is the direct copy of the gene you want to express from the nucleus, from the DNA. It can, so mRNA copies can translocate and be like allocated in the synapses at the bottom of them. And so this means that for very fast and rapid signals and communications, the synapses can by themselves, without too much of a contribution of the nucleus, respond to this activity, local activity, and get strengthened, become bigger. So this is very rapid and very efficient for like, for example, also for these short-term forms of memory. So whenever people have to remember a number for a few seconds, this is what would happen. You have like this brief strengthening of that specific circuit that will help you retain that information, but eventually forget about it in 2 or 3 minutes time. Whereas anything that involves the activity of the nucleus has to derive by a very strong type of synaptic inputs. So this is why whenever we talk about long-term memories, we really have to have a strong signal that from the synapse already there starts all the journey back to the nucleus. And this is done through a series of molecular cascades. So usually many different receptors at the synaptic level are involved. For example, we have these NMDA receptors that would be among the leaders in these calcium cascades, and like secondary molecular cascades that will fall onto protein that are really good, like this CAM kinase protein class is really good in shuttling from the periphery of a cell, so either from the dendrites or from where the activated spines are, they can travel up to the nucleus, and they follow these crazy highways, like this acting cytoskeleton that will guide them until the nucleus, where they will induce changes, being like activating other transcription factors, gathering them, and then binding the DNA. And only there you have all these changes being in acetylation, in methylation, phosphorylation, and so on. So I only focus on acetylation, but what I can see is that some promoters will be more acetylated, and therefore the transcriptional machine can be locked into this acetylated site and start its journey along the gene body. And when we will have this new copy of mRNA for all the genes that are needed to support the new formation of spines, or the new formation, or maybe just an enlargement of the we call it this dendritic spine, then all this messenger RNA will be transferred to the cytosol, where it will become translated into proteins, and these proteins from there will start their journey, as you said, towards the periphery again. And what I find always interesting is that also in this journey, it's not really yet super clear what tells a protein to stop there. So there must be signal as well involved in this that can really guide this trafficking of proteins and enzymes all around to the right spot, to the spot that needs to be to be strengthened. So I, in terms of how many molecules of ATP this takes, I cannot tell. I haven't measured them all, but I am pretty sure quite many.
It's probably quite expensive.
Yeah.
Yeah. So, I mean, if people think about it, right, in our own body, there are neurons that are, meters long. There's neurons that go all the way from your head down through your spinal cord. So there can be quite a distance that has to be traversed by some of this machinery. But it sounds like what you're saying is, roughly speaking, there's some short-term things that can happen and some long-term things that can happen. So at many synapses, you've got mRNAs that are already at the synapse. You've got ribosomes that are already at the synapse. All of the stuff needed to turn the
Even
mRNA into a
mitochondria. So
yeah.
this ATP sources, mitochondria are also at the synapses.
Yep. So there's mitochondria. There's basically everything the cell needs to turn that mRNA into a protein and stick it somewhere can already be there at a synapse. And so that can help mediate some short-term things. So if you imagine, I think you used the example of just remembering a phone number briefly, everything you might need to do something like that in the short term is already there oftentimes. But then also there's, as we all know from experience, we can encode long-term memories that we store for weeks, months, years. And these longer term changes often do require communication between the nucleus and the synapse. And so there are mechanisms in place, even if we don't understand what they all are, that allow some of the short-term stuff to happen and some of this long-term stuff to happen that involves communication with the nucleus. When we think about strengthening a synapse for the long term, say in the service of encoding a new long-term memory, from a neurobiological standpoint, can you talk a little bit about LTP and give people a sense for the essence of what LTP is?
This is not an easy question because there is, I think, still a full debate. And I want to precise, I'm already sorry because by formation, so I might say plenty of things that are not 100% correct. So from my understanding, so what I imagine when I think about LTP, which is this long-term potentiation of a synapse, is a readout of how strong a synapse is, because and how much response it will evoke once it's stimulated. And so what we know is that, for example, this type of readout, of measurement, has been associated to memory. For a very long time, there was almost like this kind of sense that NLTP, so if one finds an NLTP type of phenomenon at a synapse, it is an indication of a memory event, because we know that memories, long-term memories, in theory or in practice, they do make our system stronger, like the memory circuit stronger. And so what we saw is that after a very strong activation of pathway, we see that the synapses involved in that pathway will be strengthened. And this means that if you go at a second time and you stimulate that synapse again, the response to the same stimulus will be much stronger. So it's let's say a synonymous of an event that previously happened that strengthened a spine that before, when you recorded, needed certain current to be activated in a certain way, whereas even a subthreshold type of activation will lead to a similar response this time because that synapse has become more strong. So this is what I imagine. I imagine all the cascade of events that in my from my field. So as a molecular biologist, I do think that because a previous event of signaling has induced a lot of changes, which mean that they led to a restructuring of the synapse, and this time the synapse will bear more channels like AMPA receptors and NMDA receptors. There will be more, like even the size of the spine will be larger to harbor more of these molecular and proteins, molecular factors and proteins that make it more stable. Then if I subthreshold, if I use a subthreshold stimulation, this synapse will be able to respond equally strongly as the first time that I really needed a lot of current. I hope this makes some sense for you too.
Yeah. So basically, if we simplify it a little bit, you can imagine a synapse receives a message. That message can be relatively weak. It can be relatively strong. And when the message it receives from other neurons is particularly strong, this can activate that synapse in ways that it is not activated when a weaker signal comes in.
Exactly.
And this involves a variety of things, including calcium influx into that synapse. And like, what are some of the hallmark things that happen when you get a very strong stimulus, something that's capable of inducing LTP, that's capable of making that synapse bigger and stronger? I know that one of the things that happens is some special genes get ultimately turned on. And so what are some of those things that happen in response to those very strong stimuli all the way back in
the so as I said, like these very strong stimulations will induce these secondary messages, like they will induce this cascade that will activate the genes. And so this means that it's not one gene, one LTP. So, but however, there have been, like, for example, hubs of genes that were mastered by some transcription factors. And among, like, the most famous in the learning and memory field, there is this CREB, which is like it's like a CRE binding element. So this transcription factor can recognize some element of DNA and bind to those elements. And these elements are found in the proximity of the genes, of some of the genes that are really important to support this, like synaptic activity and transmission. So here we see how much, like, the importance sometimes of one transcription factor can really have on multiple types of genes that are all necessary to support a stronger activation. Mm-hmm. But LTP, for example, has been challenged by using protein inhibitors, for example, or transcription inhibitors, and in both cases have been found to be like defective. So if you inhibit the transcription of genes or the translation of proteins, then you cannot have the LTP, which really speaks in favor of this fact that you need this communication from the synapse to the nucleus and back in order to have this potentiation of that synapse.
Mm-hmm. So it sounds like when you have a very strong input stimulus that can stimulate LTP, there is a signal that's created that leads to some coordinated changes in gene activity. So mm-hmm. an individual, the example you gave was CREB, C-R-E-B. There's an individual protein that can turn on multiple genes at the same time. And mm-hmm. I would imagine that the reason you have that kind of coordination is there's multiple genes encoding multiple proteins that are going to be necessary for synaptic strengthening. So what are some of those genes? Are they things like ion channels that will make the synapse more sensitive and things like that?
Yeah. So I can talk about, like, for example, what we found in our study. So what I was looking for were genes that would compose somehow a signature of what would make a neuron more, like, easily predisposed to become part of the memory network. So what we thought was, okay, if acetylation is important for gene expression, and if memories are, like, long-term memories are really, they necessitate protein expression and gene expression, then what if there is, like, this connection between acetylation and memory formation? What if the neurons that have to become part of the memory network have a higher content of acetylation already as a baseline compared to other neurons? And what we saw is that whenever we manipulate, so we artificially increase the acetylation levels of random neurons, that, of course, we pick regions that we know will be important for the memory networks we want to investigate. Then, whenever we increase the levels of acetylation, we see that no matter how, so by using different types of enzymes that can do this, that can mediate this increase in acetylation, we see that the DNA regions sensitive to these increases in acetylation are highly similar. So that speaks in favor of the fact that these variable regions of the DNA that can be found either in closed or more open position, they are very conserved across different neurons, because no matter how globally you induce increases in acetylation, the specific regions that will be sensitive to it and therefore become more open will be always the same. And as you said, these regions, what we found was very interesting, because indeed they are related to potassium channels because we know potassium channels are really important in the rectification of the action potentials. So, and which means that essentially they contribute to the frequency of the spikes of a neuron. So potassium channels are able to give a pace on the action potential. So these firing events that happen in neurons once they are stimulated. And in the context of memory, we can think this phenomenon being very, very relevant, because of course, if a neuron is more sensitive and sense the environment, the stimuli, the incoming stimuli better, then the possibility of spiking and the intensity of spiking will be higher. And for memory, we can imagine, okay, it's really important if we want to encode a random event that our neurons are ready for it, are ready to activate all these processes that eventually will lead to the storage of the specific event we're talking about. So, I see. So coming back, yeah.
I was just going to say, okay, so part of the puzzle here is there are many neurons in different regions of the brain that could potentially become part of a memory, but not all of them do become part of any given memory. So the question is, well, why is it some neurons but not other neurons? Before zooming back into the molecular and cellular level, I want to zoom out and just give people a sense for the system you're working in. So when you're talking about memories and you're talking about these experiments, what's the experimental system? Like, what animal are you using, and how are they being made to remember something?
Yeah, so we use mice. So we use these black 6 mice, which are lab mice that we know are very able to form memories, because animals, I mean, in general, mice, they have to learn a lot already in their wild environments, right? So they have to know how to search for food. They have to remember where there was a danger, a predator. So they recognize smells. So their memory system is quite developed. And so we used, in particular, we kind of made usage of the fear memory network that mice have, because, as we said, they have to respond to fear. They have to be scared of predators, because in that way they increase their survival rates, right? If you are completely negligent to danger, then your success rate drops. So the fact that the mice can learn what's scary, what is not scary, and how to search for things implies that they have a very well developed and defined memory circuit. And there is plenty of studies really dissecting exactly which regions in the brains are important for the different aspects of memories in mice. And we use in particular this fear memory network, and in my case, an auditory type of fear memory network, where we use like this very traditional Pavlovian type of paradigm, where a food shock, in my case, very mild, because I wanted to use sometimes you just want to make sure that your mouse remembers something, and therefore you might use very well established protocols that you are 100% sure will elicit a behavioral response that we can translate into a yes or no type of output in yes, the memory is there, or no. And in the context of fearful memories, the freezing is one of the types of behaviors that is very easy to detect in mice. And therefore, there isn't too much interpretation from the experiment. Nowadays, we can even use like this machine learning type of strategies to be even more blind to the type of scoring we give to mice, and make sure that it's not our bias that says yes, a mouse is moving or not.
Yeah, but in this case, it is very obvious, right? Like if you've seen this with your own eyes, if you pair the tone, the auditory stimulus with the shock to a mouse, they will freeze, and it's quite dramatic, the freezing that they do.
And it's instant. And so as soon as the tone is played, then you really see that the mouse first was chilling, sniffing around, and then it really stops and kind of also like closes up and not hides, because sometimes there is no space for hiding, but really goes more to the periphery of the chambers where we usually run these experiments, and stays in the corners. That it's also quite a sign of of danger and of being scared.
Mhm.
And yeah. And so in my case, I use this milder version where let's say the US, we call it the unconditioned stimulus. So this shock was not obviously extremely dangerous. Of course, we wanted to have a platform where we could see, but when playing with acetylation levels, whether this would change the salience of the memory. So if this acetylation had any impact either on increasing the memory, making it stronger in the expression type of behavior, or maybe weakening the memory. And so, but the procedure of the protocol was the same. So these mild shocks were associated to sounds on day one. And then on the following day, you change the setting to make sure that it's really like a type of auditory memory and not like a contextual memory. So you change the chamber, you change the odor, you change the shapes around the chamber, so that then by playing the tone again, you check whether the mice recognize it or not by changing their behavior and increasing or decreasing their freezing.
And in which part of the brain are you looking at here? Where is this stuff happening?
So we used as a target this region called lateral amygdala. And amygdala in Greek it means like almond. So because it's an almond-shaped type of region, we have two amygdalas in our brain, and so do mice. And they are really like at the bottom, lateral bottom part of their brain. And this region has been shown by many brilliant studies to be really fundamental in the formation of these auditory fearful memories, because it's the recipient of auditory projections, so auditory inputs that come from either the auditory cortices or the thalamic auditory in neurons, and therefore, and at the same time, it also receives this painful, like dangerous type of signaling. So it's an incubator, and it's this associative hub that by receiving both inputs, auditory, so the stimulus and the painful stimulus, then it can combine both stimuli in neurons that will now be able to respond even to only one of them. And this is why we say that here there must be some type of potentiation, because at the time of the recall, so the next day when the mice are probed for their memory, what we do is to only play with one of these two stimuli. We only induce a response of the auditory cortices or these auditory inputs. So there will be only that type of activation on the lateral amygdala neurons. So without no painful stimulus. And now we see that by only having that one activity, these neurons, they can still be activated again and trigger this defensive behavior, this being afraid type of behavior called freezing.
I see. So there's a part of the brain within the amygdala where there's a convergence of information. You get the auditory information that tells you about the tone that the mouse is hearing, and then you get the information about the foot shock, the physical sensation that's coming in. And then obviously these two things are being paired together. So there must be some kind of reconfiguration in this brain region that enables the animal to make this association and remember it. And then sort of connecting that to what we were saying before, there's a bunch of neurons in the lateral amygdala that could underlie this memory, but only some of them actually kind of get hooked up into the memory, so to speak. And so there's this question of why some neurons and not other neurons. And it sounded like that had to do with the acetylation of these histones and how ready they were to turn on certain genes.
This was precisely our hypothesis. So to give you a little bit of a background, I do like numbers. So it's thought, there are plenty of studies have explored this region in detail. And what they saw is that there is around 70% of the principal neurons, which means this excitatory neuronal population that can receive both these inputs. So the foot shock information and the auditory stimulation. So among all the neurons in the LA of the excitatory neurons, 70% is perfectly suited to become part of the memory network, of this associative memory network. However, when we have tools to really detect how many are actually responding and forming these memory traces, And what we see is that usually between 5 to 20% are actually recruited at that moment. So in the last 2 decades, really, like many scientists were wondering how come neurons are selected, how come only so little, and what's the rule? Is there a rule? And initially, of course, anything we don't understand, we call it random. So initially, it's called memory allocation. So before it was thought to be a random process. So randomly, a memory has to happen, and some neurons are selected. And we didn't even think it was an active process. This was a passive, random type of mechanism through which neuronal selection was taking place. However, then many groups, like, for example, Sheena Jocelyn, Paul Franklin, Dalcino Silva, really started to dig deeper into the process of memory allocation to try to understand whether it is passive or active, whether there are some rules. And what they really try to understand is whether there was a type of signature that some neurons had that made them better at becoming part of the memory network. And they found that there were 2 features that would be predisposing some neurons to be better placed for memory encoding. One feature being intrinsic excitability. So we come back to this concept of readiness of a neuron to respond to stimuli, right? So intrinsic excitability is this process, is somatic. So somatic being this body area of a nucleus. So there we have, again, channels, sodium and potassium channels that define how ready is a neuron to fire, how much current do I need to inject in a neuron to see a spike. And we see that not all the neurons have the same type of intrinsic excitability. So it seems that if a neuron has it a bit higher, then its likelihood to become part of the memory trace will be higher. And equally, again, they also tested this with CREB. So again, that important transcription factor mediating many different genes. And they also saw that CREB levels can be different, and the neurons that have more CREB levels are also predisposed for memory formation.
I see. So neurons that are more excitable, more sensitive to their inputs, to receiving information, they're more likely to become hooked up into the memory trace.
Yeah, exactly. And this for us, I mean, while I was thinking about, okay, what do I do with my PhD? I mean, what kind of project will I ever have? I really got hooked by these studies, because I really thought, okay, here we're talking about neurons that are seemingly homologous. They can be twins, they can do, like, they are like, almost identical. However, one responds better than others. And the fact that there were, like, physiological signatures such as intrinsic excitability or higher CREB levels, to me was really speaking in favor of somehow a super identity, like a new level of identity on top of being the same type of neuron in the same region, connected in the same way. You also have an extra tag, an extra type of label that tells you, okay, you're even more excitable, you're even more ready to transcribe genes with PrEP, right? So I really thought, what if this fact that you randomly have some neurons having these higher features would derive by higher acetylation levels in those neurons? Maybe those neurons, they can be more intrinsically excitable because they have a DNA conformation that is more open, and therefore that allows for the transcription and the maintenance of these higher intrinsic excitability states. So this is really the beginning of the story.
Okay, and so basically, walk us through some of the major findings here. So the idea was, okay, you're in the lateral amygdala. This is a place where we know that information converges. That's really important for memory. It's very well established that this region is important for these type of Pavlovian conditioning fear memories that you're inducing in the mice. We know that you've got a bunch of neurons there that are seemingly identical. They're the same type of neuron in the same type of place, receiving the same type of input, but only a fraction of these neurons for any given memory are actually going to be recruited into the memory. And you had these ideas around, okay, well, maybe the neurons that are recruited have their chromatin more accessible. So they're more readily able to turn on the genes necessary to, strengthen synapses and do things like this. And so
Yeah.
how did you approach this, and what did you find?
So, first of all, we had to answer a very simple question, which was, do we see that the lateral amygdala neurons have different acetylation levels? Like, do they differ in their content of acetylation? Because, of course, if you would see that all the neurons in the LA had the same amount of histone acetylation, then there would have been no point, right, in pursuing this like big research. And so we did some studies where we tested different types of epigenetic marks, some that would decode and detect like a more closed chromatin, like pieces of heterochromatin, such as HB1, which is a protein quite known in epigenetics to really identify closed area of the genome. And we also use different kinds of histone acetyl marks to really check whether it was, because as we said, histone acetylation can be very complex. So depending on which type of amino acid histone acetylation happens, then you might have different responses. So we had a look at different epigenetic marks. And this was the first time where we saw that we see different levels of histone acetylations and in general, chromatin conformation in seemingly identical neurons in a structure. So in DLA, this was the first time that we could explore the diversity of the overall content. So this was not specific to any genes, it was really like a brute force type of approach to really go and look at the abundance of some marks. Mm-hmm. And what was also interesting was that we could correlate which neurons were active after a memory encoding, which means memory encoding, we say it when we want to stop the paradigm exactly at the moment where the mice learn the association. So this is day one. The mice went for the first time in this chamber. They had to learn to associate the food shock to the tone. And immediately after, they are probed for the presence of a protein called CFOS, which is a gene that is like a rapid response genes that is really highly present in neurons that are activated at a certain time. So by correlating which neurons would be marked by this gene with the high acetylation, we could really see that after a memory is encoded, the neurons that are more active are the ones that have more acetylation.
I see. So just at any given time, you sort of look at the acetylation levels of neurons in this part of the amygdala, and it just happens to be that some cells have more unwound available DNA, some cells have less. So then you induce the memory, and right at the beginning of the memory, you look for the signal, this gene transcription signal of plasticity, basically, and you find that, okay, the neurons where the signal turns on also tend to be the ones that just have more open and accessible DNA.
Yeah, wonderfully said, exactly. And this was a fantastic entry point for us because we could say, okay, at least at the correlative level, not only do we see diversity and heterogeneity in the acetylation profile of excitatory neurons, but we also do see that there is this correlation, which was exactly what we wanted to test, right? We really wanted to now move from a correlation to more of a causation type of approach, where we could assess whether by overexpressing the enzymes that can increase the levels of histone acetylation, we would see that only the neurons in which we induce this increase in acetylation, those would be the ones that upon a certain protocol for memory formation would become the one harboring the memory trace. Mm-hmm. So the experiment now went from an observational type of approach to really a more experimental, and we manipulated the system because we went into the amygdala of some mice. We only sparsely modified some of the neurons that we wanted to see whether they would become more like prone to be part of the memory trace. And afterwards, so once we increased the levels in a sparse population of neurons, we could check, okay, if we now subject the mice to the usual memory protocol, what does happens to those neurons? Do they become part of the memory trace more or not? And what we could see is that we would really like compare to chance levels, because these experiments are always done with like a control where you just kind of do all the same procedure, but instead of changing the acetylation, you just add like a fluorescent protein that just tells you that that neuron was touched, essentially. So compared to this control group, we double the chance of neurons to be selected by just playing with the acetylation.
I see. So you're able to inject something into this part of the brain sparsely so that there's an increase in the number of neurons that have chromatin in the open configuration. And you find that when you do this, those neurons that you end up manipulating are more likely to be recruited into the memory, basically.
Yeah, precisely. And what we wanted to do with that was also to make sure that I said sparsely for a reason, because if we would just float the overall structure with more acetylation, you couldn't really see like the network changes within the same animals, right? Instead, by sparsely playing with only a few, then you can kind of keep your manipulation consistent to this 5 to 10% of neurons touched, which is like the same percentage of neurons that usually shine up upon a memory formation event.
Yeah, I mean, I would imagine that might also result in memory changes above and beyond what naturally happens. Like if all of the neurons get recruited, maybe the animal, I don't know, maybe it can't remember it as well, or maybe it remembers it forever,
I mean,
or
that's a
something.
fair point. There are like plenty of studies. For example, you can play with epigenetics also using some drugs, right? So there is like it's also an FDA type of drug, like approved drug, the HDAC. So HDAC is this class of enzymes that removes acetylation. And if you use inhibitors for this type of class, which implies that then if you remove or inhibit the enzymes that remove acetylation, the outcome is that you have higher acetylation levels. So this is global in the whole brain. And yet you see that the mice tend to have a little bit more improvement in memory retention. So I think the brain is great because it can always compensate somehow, even intrusions, So who knows if we would plot the overall system, who knows what would happen. By this study, we see that it's not so easy to disrupt brain activity and brain functioning.
And so, okay, so you can render a subset of neurons in this part of the brain more likely to be recruited into the memory by changing acetylation, by changing how open the DNA is inside of those neurons. And then you see that they are recruited into the memory, basically. What about sort of the other side of this? Can you disrupt those neurons and get rid of the memory?
So we did several things to make sure of how things would work. So we did, like, I would like to mention a couple of experiments which I think were really nice. So we did, because I told you, right? So you can increase acetylation if you increase the amount of enzymes, which are called HATs, so histone acetyl transferases, that do this type of job, right? So by increasing the enzymes, you would then in turn increase the acetylation. But these enzymes can do so many things. So they can bind other proteins, that they can go around. So we didn't actually know whether what we were doing was truly only to play with acetylation. So what we did was to use, it's called dead mutants. So we took the same enzymes and we mutated specifically only the region that can acetylate histones, which means that those enzymes were like capable to do everything else. And when we do that, we saw that that memory allocation was not being predisposed. So those neurons, this time having these mutated enzymes, were not becoming more part of the memory trace. So this was one evidence really telling us that it's necessary to see that type of change. It's necessary to have the catalytic domain that is involved in the addition of the acetyl group in these enzymes to make a neuron become part of the memory network. Also, what we did was to overexpress the counterpart, so this HDAC enzymes. So by overexpressing HDACs, so by in principle decreasing the overall global acetylation levels, we did not see any change. And therefore, again, the system seems to rearrange, not really care what we did. And I think really wonderfully what we saw is that if you then endogenously repress these enzymes using these SH short hairpins RNA, which are tools where you can target the endogenous enzymes, mRNA, and you can suppress it. So you really remove them from the system. There you see that the neurons that receive this manipulation will not be recruited during the memory task, really saying, okay, you really need to have these enzymes to have more acetylation and then to be recruited.
So by manipulating these enzymes and how plentiful they are or how active they are in the cells, that it has a direct effect on the memory side of this.
Exactly. Yeah. So
you guys can do this experimentally, obviously, as scientists. The levels of these enzymes, the acetylases, the deacetylases, the things that control how open or closed the DNA is, and therefore how readily neurons might participate in a memory. What are some of are there natural stimuli that change those things? So, for example, if you sleep deprive a mouse or you give it exercise or you give it a certain diet, can this modulate how active these enzymes are and therefore how readily learning and memory will happen?
Yeah, I mean, this is a great question. I think it's a composed type of approach to this, because for sure the enzymes levels oscillate, and there are plenty of, as you mentioned, a few, right? Exercise, for example, is one. Sleep deprivation also has been linked, or even like aging by itself, like all these phenomena that happen throughout aging or dementias, they do have an impact on the levels of these enzymes. Some cognitive pathologies, like Huntington diseases, for example, is linked to the differential expression of levels of some of these enzymes, because these classes of enzymes, they are composed by many members. So even if you have a lot of different members of the same class, but you don't have one of the members, this can really have detrimental effects for the system. So for sure, the levels of enzymes are critical, and there can be genetic reasons why you have different levels. There can be also environmental reasons, both internal environment as well as external environment. But at the same time, these enzymes will not work the same if they don't have the chemical groups to act on. So not only is important to have the levels, also how much material they will have at their disposal will influence what can they do, right, inside of a cell. So it's really like to really study this type of processes is complicated, because I think it's beautiful at the one hand, because this is really like this one sensor that can be really like understanding so much of what's going on around us. But at the same time, it's also complicated to address experimentally because of the variety of signals that can disrupt or change their activity.
And so you have this chromatin plasticity that's necessary to recruit neurons into a memory. How much do we know about how the memory actually gets stabilized? So you have to have DNA unwound in the nucleus. It has to be able to be transcribed. You then have to have the requisite stimulus received by that neuron to start transcribing these genes. But then the memory also has to be stable. So what do we know about how the cell regulates gene expression so that you turn on genes that are needed to create the memory, but then you prevent it from going away?
Yeah. So I think a quick answer to that is like, to make sure you're going to recall your memories, it's a great tool to remember better. And this is why we need repetition, right? Because we need to strengthen these storage events. At the same time, it's also interesting that every time we remember, these traces become labeled again. So which means like there is a sort of malleability to these activation events of these memory networks, because we can assume that, okay, a neuron gets activated when we encode any type of information. So, for example, me and you are having this chat. So right now in my brain, some neurons are on, and gene changes are happening if I will store for long term this moment, which I think I will. But this means that if in a month I will talk to a friend and remember this moment and tell, okay, he asked me this question or this other question, any perturbation at that time will become part of my memory in itself, because while we remember, we are still exposed to a new context, to new questions, to a new environment. And so we have observed that anytime we recall this opportunity, this window of opportunity to strengthen a memory, because recalling is reactivating and therefore going to the genome, reactivating the machineries, re-putting new proteins and stuff, because we are telling our brain this is important, we access this information, so let's make it stronger again, could also be subjected to deformations somehow, to enrichments or like embellishments or to maybe disruption if we do something else, like we can use some drugs that at that time will be inhibiting our protein synthesis, and therefore we will facilitate the forgetting of this memory too.
Mm-hmm. And, we mentioned earlier that all of this stuff is very energy intensive. There's a lot going on for a neuron to participate in memory encoding. It's got to make new proteins. It's got to turn on certain genes. Things have to get to the right place. There's a lot of stuff going on, and there's lots of different ways that neurons can have energy or create energy. So naturally, I start to think about things like diet or where the neurons are getting their energy from. Are there certain, like, for a neuron like the ones in the lateral amygdala that you're studying that have this potential to be encoding memories and participate in this memory trace, are they like particularly good at creating ATP? Do they have more? Do the more plastic or more eligible neurons have more available energy? Do we know anything on that side?
So I've never read like about precise studies. There might be, but yeah, if there are, like we can have a conversation once I read them. But on the precise mechanisms, or if some regions are much better at processing. For example, what we know for sure is that neurons use glucose and lactate to produce energy. And what is interesting is that like 20% of our energy consumption happens in the brain. So the brain is really like sucking up a lot of glucose and converting it in so many different metabolites and compounds that are necessary for its performances. And indeed, if you don't have energy, you also see that your cognitive abilities you're not as sharp, you're not as quick. So it also is interesting how much other cell types that are found intricated among the neurons, like astrocytes, really infuse nutrients like lactate to support the metabolic content of neurons. So it's not exclusively glucose centered, but the energy supply of neurons is again, I think, very rounded. It's very robust the way neurons get energy going, and they don't do the job all by themselves. There are other cell types that support them in that, and I think this is also beautiful because it really talks in favor of this sensing, and maybe by playing with acetylation, we are also playing in the with the way neurons communicate to other cell types to maybe change the way they get different nutrients in order to then produce the building blocks necessary for their functioning, essentially. But this, I think, would be wonderful to study exactly what type of energy is necessary in different brain regions or for different tasks even, right? Because maybe memory is one of the many things that the brain does, but maybe for doing more type of maintenance, type of brain activity, you don't you might not need the same type of energy you need for a memory event to take place, because some processes are more long-term, they are slower, some others instead are quicker, and therefore also the type of energy supply could depend based on the type of task that the brain needs to do.
Mm-hmm. And so you guys identified this mechanism of chromatin plasticity that predetermines whether or not a given neuron in a particular region becomes part of the memory. It has to do with the acetylation levels present within that neuron. So the neurons that were sort of readier to transcribe certain genes because their chromatin was in a certain configuration are more likely to participate in the memory. We know that those neurons actually do participate in the memory, and you guys can disturb them and show that the memory is disrupted. What are some of the next questions here that are implied by your study, and some of the things you guys might be doing to follow up?
Yeah, so I mean, I think one of the main results of these studies was given by this multi-omics experiment. So we did this experiment where we decided to immediately after our manipulation, in the absence of any learning paradigm, we decided to extract and analyze the neurons we manipulated to understand how our manipulation was affecting the chromatin in those neurons. And what we saw was that there were some regions of the DNA that became more accessible, others that instead closed up. And there were even yet another class of regions that were already accessible at baseline. By just adding more acetylation, they became much more transcribed. So, and this is like a very diverse type of information we could gather, because we could understand that a DNA is really built in a way whereby acetylation, which is one type of manipulation, can have different outputs. So how can this happen? Why one manipulation can lead to different outputs? And what does each of these outputs mean in terms of memory formation? So for example, what we assumed is that because we saw, we kind of stopped at the very early phases of memory formation, right? We just, as soon as a memory event happened, we stopped our manipulation there to see what we wanted to observe, which was, okay, how does this mechanism of memory allocation works? And for that, we assumed, okay, maybe since there were genes that became more transcribed, so most likely proteins that were more expressed already from a baseline, from the baseline, those genes are potentially the ones that make those neurons become more more predisposed. But what I wanted to say next was that, however, we also noticed that upon this change, upon this increase in acetylation, not only we saw an increase in neuronal predisposition, but we also saw that remotely in time, the mice in which we induced these manipulations could remember better, could remember more long-term. So our assumption is maybe all these other regions that changed only the genomic architecture, but not their transcriptional expression from the beginning, maybe those regions are sensitive to late type of changes that have to happen for maintaining, consolidating, strengthening the memory long term. So, of course, we don't know that yet. This is our hypothesis right now, but really examining how these two classes of genes that changed in different ways might contribute differently to different aspects of the memory timeline. This would be one of the most immediate follow-up to really dig deeper and understand the molecular codes of the different phases of a memory. So,
yeah. So obviously you guys observed a set of changes, but this is sort of on the short-term memory side, or the early side of the memory.
Yeah.
And
we call it recent memories. Yeah.
Yeah, recent memories. So if you were to continue exposing an animal to these stimuli and it really, really remembered things in the long term, the idea is maybe repeated exposures and repetition that's going to result in a very long-term, highly consolidated memory. Maybe that recruits additional parts of the genome and additional types of proteins that are responsible for that later phase of remembering.
Yeah, this is one possibility as well. Like also, what I am very intrigued to check is, for example, we know that some pathologies, let's take Alzheimer, for example, we know that they are associated to memory disabilities. So one of the question could be, like, we know that Alzheimer has been seen as not only a neuro a neuropathology, but it's right now there is also this new type of vision where we think of Alzheimer also as a metabolic pathology. So there is insulin, like they call it diabetes type 3 as well. So it's interesting that there is for a dysfunction of the brain associated metabolic dysfunction. So one possible idea is also to go and test our same hypothesis in models of Alzheimer and see, okay, now that we understood that in naive mice, we see this correlation, do we see differences in Alzheimer models? Do we see differences in as a result of increasing histone acetylation levels, for example?
Mm-hmm. Yeah, it's also interesting to think about things like the distinction between retrograde and anterograde amnesia. Some people have short-term impairments where they can't encode new memories. Some people have long-term impairments where they forget things that have been encoded for a long time, but they can form new memories. And so it's interesting to think about all the potential there. Like maybe there's different proteins involved in different aspects of this epigenetic regulation, and you can disturb some of them to result in, problems on the initial encoding side, and maybe you can disturb other ones that give on the consolidation
and
side.
For example, as a follow-up to this point, it's also interesting that they that different groups, research groups, explore different chemical groups, for example, histone methylations. And they saw that, for example, during memory formation, if you play with the histone methylation levels, that doesn't really change things much. But for the consolidation of the memory, for a better recall, then it's where it really makes a change. So on top of what makes really memory be more resistant or less resistant, more prone to forgetfulness or not, it also maybe there is also a link with how much of each of these type of epigenetic marks there is and at what time in different neurons. So the complexity is really exponential, but I think it opens up so many new approaches that we could take on these features of memories.
Mm-hmm. So what's next for you? It sounds like you're doing some follow-up experiments now, but do you think you're going to continue on sort of this line of research or jump to something different?
So I actually so I'm saying it to you for the first time. But I accepted an offer, a very exciting offer for a postdoc in Fides Zenk lab, and she's studying human brain organoids. So my interest is now to move to a very different system, which is this human brain organoids, and check how much epigenetics and neuronal function at this very primordial level, how much these functions that I've observed in a context of a very mature, well-developed system like the mice, can we find that even in human brain organoids, epigenetics and neuronal function, they depend on each other. So I really want to kind of maybe one can see it as a step back or step forward, depends on where you want to look at, right? But if you take an even simpler system that is still developing, do we see in human cells that are reprogrammed to form brains, that by changing epigenetics landscapes, we do change the way these organoids would form, either for the worse or for the better, really to dissect what type of modifications contribute to which kinds of functions. And what I would really like as a layer to this is the contribution of metabolism, as we said. Mm-hmm. I had a lot of fun working with mice because it's great. You can see with your own eyes how much you change the system. You can explore the behavior, you can explore their cognitive potential. But the limiting side of it, which I think is a good thing, is that you can't really use that many because it's a very important use of animal models. And for research, I mean, of course, we're curious beings as humans, but we also have to really stick to precise ideas and not waste too much material. So we have to be very specific in what we want to do. And the good part of working in vitro sometimes is that you can have much more material to play with, and you can go very deep because technologically you need to go very deep. You need a lot of material, and therefore I think this type of system would really allow me to explore and deeply investigate the connections between metabolism, epigenetics, and neuronal function in a way that would not be possible to do right now in mice.
Well, Giulia, this has been fascinating. Is there anything that you want to reiterate or any final thoughts you want to leave people with in terms of the neural basis of memory and its relationship to epigenetics and chromatin remodeling?
Yeah, I think as a take-home message, what I would like to share is this new, like I think we need to be a little bit more open, like our chromatin, when we think about memories, because right now we have on the one hand electrophysiologists and on the other one cellular biologists, and we think that either everything depends on synapses or everything depends on the nucleus, and I think that absolutes never work, and we need to really find a middle ground. And I find it beautiful that we can see how much these two aspects of a same cell influences each other. So even memories that seem these very big microscopical processes that truly depend on synaptic connections, because of course, if we would disconnect our brain, we would never be able to perform any type of activity. But this connection really has to happen in specific ways that depend on the way the nucleus instructs every cell to become what they have to become and to perform and function in a suitable way for what their future possibilities will be. So it's not all epigenetics, it's not all connectivity and functionality, it's both. They have so the chromatin and its outputs and the neuronal outputs have to talk and really define each other at any given time for us to work at our best. And I'm very proud of our story because I think it's one of the first steps towards this direction.
Yeah, no, it was a really nice paper and really good work. So I look forward to seeing what you do in the future. And thank you for sharing.
Thanks for having me.
This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure. And vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.